Hey students, let's get started with our discussion on the reproductive system. Hopefully I'll be able to squeeze this body system into 3 lecture recordings. I'm shooting for about 20-ish slides per recording. And without further ado, let's get started. So, what on earth is going on with our bodies? We need to have a way to reproduce, to repopulate our species, to create more progeny. And there's a problem in that we have our genetic information contained in deoxyribonucleic acid, DNA. We need some way to repackage our DNA from our parents to our generation, to our children's generation, to the next generation. This is how we have the continuation of our species. And there's a big problem, a big catch-22. If we were to just take 2 regular human cells and combine them together, we would double the number of chromosomes in that cell. Each body cell has 46 chromosomes, 23 pairs, or 2 sets of chromosomes. And if we were to take a regular human cell and combine it with a regular human cell and then produce babies with it, we would have 92 chromosomes in each of those baby cells. And then if those babies reproduced this generic way, or just take random cells and combine them together, they continue this doubling of the number of chromosomes. If you're familiar with trisomy 21, Down syndrome, or trisomy 18, or other diseases of the genome, you've probably noticed that if you have extra chromosomes or missing chromosomes, that's a really bad thing for you. That's a great way to die or have severe cognitive and physical defects. So we need some way to closely monitor the number of chromosomes and to make sure that the number of chromosomes in the human species as a whole remains constant from generation to generation. And here's how we solved it. We are going to cut the number of chromosomes in half with a select number of cells. Most of the cells of our bodies have 2 sets of chromosomes. They are di for 2, diploid in nature. However, we can make gametes or sex cells. These sex cells have only 1 set of chromosomes. They have 23 total chromosomes, 1 chromosome of each pair in their nuclei. So regular body cells, these are called somatic cells, they have 46 total chromosomes, but the sperm and egg have only 23 chromosomes. These cells that are in our sperm and eggs, these gametes, are going to be haploid in nature. When I say haploid, I mean they have only 1 set of chromosomes, abbreviated as just a lowercase n. Regular body cells, somatic cells, are diploid, which is abbreviated as a 2n. When we fertilize an egg, so when a sperm and an egg come together, or secondary oocyte come together, that single cell that has a unique human genome is called a zygote. And this zygote will have 46 total chromosomes. So we start out as a mom or a dad, and we start off with 46 chromosomes in our bodies. Then we chop the number of chromosomes in half to make our gametes, or sex cells. And then when our sex cell combines with our partner's sex cell, we will take 23 plus 23 and have a child with 46 chromosomes in the cells of their body, so on and so forth. And by cutting the number of chromosomes in half for our gametes, we can ensure that we have a constant number of chromosomes within the human species as a whole from generation to generation. When we just divide and reproduce a regular cell, we go through a process known as mitosis. This is the duplication or division of the cell colloquially. If you take AP Bio or human anatomy, or the anatomy and physiology courses, we get to be a little bit more anal retentive and say mitosis is the duplication of the nucleus, and then cytokinesis is the duplication of the cytoplasm. But for this course, we're just saying mitosis is the duplication of the cell. We start off with 2 sets of chromosomes in the parent cell, and each daughter cell will have 2 sets of chromosomes. This is the process that we go by to make more skin cells, more liver cells, more muscle cells, more eyeball cells, more of every tissue cells of every cell type in our body, except for eggs and sperm. So this is a very common process in our bodies. Now when we look at meiosis, instead of starting with a haploid cell and ending with haploid cells, instead of starting with diploid cells and ending with diploid cells, like we did with mitosis. We have a diploid parent cell, diploid daughter cell. During meiosis, we start with a diploid parent cell and end with haploid daughter cells. And these daughter cells, these gametes, are going to also be more numerous. During mitosis, we start with 1 parent cell and with 2 daughter cells. During meiosis, we start with 1 parent cell and end with 4 daughter cells. Meiosis is a process that we use to make our sperm and our eggs, or our gametes. So to do a side-by-side comparison, we can, making sperm or making eggs, spermatogenesis is the creation of sperm, oogenesis is the creation of eggs. With spermatogenesis, we start off with one parent cell, and we end up with 4 evenly sized sperm cells. So it's that one large parent cell is divided equally among the 4 sperm cells. During oogenesis, when a woman's body or a female's body is making an egg, it's going to be a more complicated process. The egg is a big, complicated cell compared to a sperm. A sperm is just a little suitcase that carries some DNA, just a delivery boy, it's errand boy. All it's doing is delivering DNA to the woman's oocyte. That woman's oocyte, though, needs to do a lot more. This oocyte does most of the heavy lifting. This egg is going to have to contain all of the organelles and the mitochondrial genome and the woman's contribution as well, the 23 chromosomes from the woman. So during oogenesis, when we have meiosis making an egg, 3 of the 4 daughter cells are going to be thrown away. These 3 over here are called polar bodies, and these are just disintegrated in the woman's reproductive tract. This egg, though, is going to be a large cell, much larger than the sperm. This figure is not to scale. We have some other photomicrographs later in this presentation I'll show you. They'll give you a better idea. So as human beings, the moms, the dads, the babies that you see running around, you can see that we all have 2 ends. We are all going to be diploid organisms. So we have a diploid dominant generation. We have a small stage where we're haploid, and that's going to be when we're as a sperm or an egg, or sperm plus egg separated from each other. We spend the vast majority of our life cycle, starting from the zygote to the grave, as a diploid organism, and the vast minority as a haploid organism. The process that takes us from a diploid organism to haploid is meiosis, and to go from haploid back to diploid, we go through the process of fertilization. So, guys and gals, why is it important for the number of chromosomes to be halved in gametes before they are combined? Go ahead and pause the video and get me an answer. You can check your textbook, check your notes. I would actually recommend rewinding the video to the first section of it. 5, 4, 3, 2, 1. It is important because we want to keep the number of chromosomes between generations constant. If we didn't have the number of chromosomes when we made our gametes, when our gametes combined, we would have a constant doubling of the number of chromosomes in each subsequent generation. Moving on, let's talk about the male anatomy. Male anatomy is simple. When it comes to the male reproductive system, it's all about making some secretions. There's not much to say. I remember when I was in grad school going through my anatomy and physiology sequence in grad school, my textbook had over 3 times the number of pages dedicated to the female reproductive system compared to the male reproductive system. The take-home message I got from that is, well, females are more complicated than males. And I'll freely say that as a dude, that I'll admit I am the less complicated sex or gender. I'm just a Neanderthal compared to my wife. Now, as I look at the male anatomy, and as we look at the male anatomy, we find that there's lots of different glands and tubes. And they come in different numbers. They either come in pairs or as singles. So, for example, we have 1 scrotum. We'll have 2 testes, or testicles for plural. We have 2 epididymis, 2 vas deferens, 1 urethra. Then we have some special glands, such as our bulbourethral, our prostate, and our seminal vesicles. And then we have 1 penis. Let's go ahead and look at the mid-sagittal slice of our male reproductive tract. I'm going to start with the path of sperm travel. We're going to start with the testes. This is where sperm are made. Sperm travel to the epididymis, where they are maturated, and then travel through the vas deferens, which contains our which is part of the spermatic cord. And that vas deferens merges with the seminal vesicle. We have 2 seminal vesicles, 1 for each vas deferens, and that seminal vesicle is then going to travel, or the sperm will then travel through the seminal vesicle through the prostate, and there's a special tube that they travel through. This is called the ejaculatory duct. This ejaculatory duct merges with another tube that carries urine. This tube that carries urine is called the urethra, or the prostatic urethra to be more specific. And the prostatic urethra and the ejaculatory duct merge to form a common pathway. That's just collectively known as the urethra. Every once in a while, if you're looking at a more in-depth textbook, it'll be called the penile urethra right here, and then the membranous urethra for that little tiny section right there. This large circular gland stands alone. It is the prostate. Males have one prostate gland. Notice how the prostate gland hugs the urethra and is directly beneath the urinary bladder. Merging with the penile urethra, the portion of the urethra that's embedded in erectile tissue, we have two more glands known as bulbal urethral glands, or if you're a crusty old dude like me, you know those as Cowper glands. And those Cowper glands are going to give more secretions that combine with the forming semen that will then exit through the opening of our urethra. I remember this little structure here, this little opening, is the navicular fossa of the urethra. It was very difficult for me to find that when I was in grad school. That's a tough name to dig up. The penis is the part of the male reproductive system that sticks out. And our penis is going to be covered with the penis will have a very sensitive structure at the apex or tip, the distal portion of the penis, known as the glans penis. In uncircumcised males, the glans penis is covered by a foreskin. In circumcised males, that foreskin has been removed. Here is a frontal view and kind of a coronal slice through our male reproductive tract. We have two testicles, or males have two testicles, two seminal vesicles, two vas deferens, two seminal vesicles, one prostate gland, two itty-bitty bulbal urethral glands, and one penis. What's the purpose of that scrotum for the male's reproductive system? The scrotum contains the testes. It's important in fertile males that the testicles descend and move down into the scrotum. Why is that important? Our testicles need to be air conditioned, so to speak. They need to be cooled off to less than regular ideal body temperature. They function optimally at about 95 degrees Fahrenheit or about 35, 36 degrees Celsius. And that temperature, that slightly cooler temperature of the scrotum, allows for optimal generation of sperm. I remember my freshman year of college, I had an opportunity to attend a seminar that was given by a sexologist, and he recommended very strongly that if you're a gentleman struggling with infertility issues, that you should never put a laptop on your lap. Most people are using tablets now anyways, but that heat generated from the laptop computer could contribute to infertility problems. He also recommended icing your testicles and wearing boxers as opposed to briefs to help decrease the amount of temperature that the testicles are exposed to. The scrotum contains the testicles or testes. These are the two male gonads. They produce sperm, which is the male gamete, and they also produce male sex hormones, most notably testosterone. This testosterone is going to be produced by the interstitial cells, and we have another slide coming up showing this better. Actually, let's just skip ahead. So we can see in the testicles that this testicle, which is kind of a bean-shaped or oblong-shaped sphere, for lack of a better term, there are many, many tiny little tubes. Each of those little tubes is a seminiferous tubule. And if I zoom in on a cross-section of a seminiferous tubule, I can see right here that in the center of the seminiferous tubule, you can see many, many, many little sperm cells forming. These sperm cells in the center of the seminiferous tubule are immature sperm cells. Sperm cells within the seminiferous tubules are made by Sertoli cells. And then in between our tubes, you can see this cross-section, this circle right here, the seminiferous tubule. In between the tubes, we have little clumps of interstitial cells. These interstitial cells produce testosterone, which aid the Sertoli cells in the production of sperm. After the sperm are produced in the seminiferous tubules, they are transmitted or moved to the epididymis. So here's the testicles, the testicle which is filled with seminiferous tubules, and then those sperm travel to the epididymis, and here in the epididymis, the sperm complete their maturation process. The process of making sperm is known as spermatogenesis. We covered that, boom, 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 on this slide. Making sperm is spermatogenesis. Making eggs is oogenesis, boom, 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 boom. The sperm that's being produced has a technical name. Most things in the human body have a common name that most people recognize, and then the hoity-toity technical name. The fancy-pants name for a sperm is the spermatozoa. Our spermatozoa will be stored within the epididymis and then released to the rest of the reproductive tract during ejaculation. As we look at a sperm cell, one thing that is notable about a sperm cell is it is the only human cell that contains a flagella, or a tail. This tail of the sperm is the flagella of the sperm. And as we look at the sperm, there's the head, There's the midpiece, and then there's the tail or flagella. The head contains a very large notable structure known as the acrosome. This acrosome is going to be the ordnance, so to speak. This is what allows the sperm to penetrate the egg. Within the head of the sperm, there's also going to be the nucleus. The nucleus is going to contain the genetic information that is being transmitted by the sperm. The midpiece of the sperm has a high concentration of mitochondria. These mitochondria function to produce ATP that power the tail, power the flagellae. You can think of the sperm as a glorified delivery boy. There's an acrosome that allows it to penetrate the egg. It has its package, its nucleus with genetic information right here. Here's the power source, all the mitochondria in the midpiece, and then here's the locomotion, that big tail. As our sperm are traveling away from the epidermis, they will travel through the vas deferens, sometimes referred to as the spermatic cord. And this vas deferens takes the sperm and transport ultimately to the urethra. Before the sperm gets to the urethra, the vas deferens will merge with the seminal vesicles, and then those seminal vesicles will send the sperm to the prostate gland, and it's within the prostate gland that the vas deferens merges with the urethra, that then transports sperm and urine out of the body. Our seminal vesicles that we talked about earlier come in pairs. There's 2 seminal vesicles, and they produce a sugary fluid that contains a lot of fructose, a lot of fruit sugar in it, and also contains a lot of citrate in it. So this fruit sugar, this fructose within the seminal vesicle secretion is what powers or what supplies energy for those mitochondria within the midpiece of the sperm. Our prostate gland is going to produce an alkaline fluid. This alkaline fluid made in the prostate gland helps to buffer the acidic pH of the vagina. As an aside, the bitter nature associated with semen is going to be primarily attributed to the alkaline fluid of the prostate gland. In general, alkaline fluids have a very bitter taste. And then if we look at the bulbar urethra, aka Cowper's gland, the Cowper's gland is going to produce mucus that is secreted before ejaculation of the male. This mucus has an alkaline nature to it, and it serves to remove acid from the urethra, and it also serves to help lubricate the female's reproductive tract so that the penis has better penetration and can reach the cervix with more ease. As we look at the penis itself within the male's reproductive tract, this is that organ that sticks out that's used for both sexual intercourse and urination. That glans penis at the tip of the penis, or the distal portion of the penis, will typically be covered by the foreskin. So here's the regular penis. It contains erectile tissues, and then at the tip it contains the glans penis, colloquially or commonly referred to as the head of the penis. And then wrapped over uncircumcised penis, there's a fold of tissue known as the foreskin. In circumcised individuals, circumcised males, I should say, most of the foreskin or all of the foreskin will be removed. Many times males will have erectile dysfunction occur. Erectile dysfunction occurs when there is not complete expansion or inflation of the erectile tissue of the male's penis. Many times this occurs because they cannot have complete engorgement of this erectile tissue with blood. To control the male reproductive cycles, we're going to have many of the same hormones involved with the female reproductive cycle. The big difference is that in the male's reproductive cycle, these hormones are only going to have one purpose. In the female's reproductive tract, depending on the concentration, the purpose can change. Both males and females will have gonadotropin-releasing hormone, GnRH. It's going to be released by the hypothalamus. That's a typo there. GnRH comes from the hypothalamus. I should really fix that typo. Sorry about that, guys. To control the release of other hormones. Actually, I'm going to fix that typo right now before I forget about hypothalamus. Save. Back to the full presentation. There we go. From the hypothalamus, it travels to the anterior pituitary gland. Well, at the anterior pituitary gland, it will stimulate the anterior pituitary to produce the 2 gonadotropins. Those 2 gonadotropins are FSH, follicle stimulating hormone, and LH, luteinizing hormone. FSH directly stimulates the production of sperm by the Sertoli cells. Luteinizing hormone stimulates the interstitial cells, also known as the Leydig cells, to produce testosterone. Testosterone itself is going to be a very important hormone for the male sexual response and male development. Testosterone will help stimulate Sertoli cells to produce sperm, and also increases male muscle mass and causes many other secondary sex characteristics in the male, such as deepening of the voice, increased body mass, increased body hair, etc., etc. So here we can see the hypothalamus. Our hypothalamus releases gonadotropin-releasing hormone, which travels to the anterior pituitary gland. That anterior pituitary gland is then going to produce luteinizing hormone and follicle-stimulating hormone that go to the testes. And then testosterone inhibits the anterior pituitary and the hypothalamus from producing or responding to gonadotropin-releasing hormone. In other words, if you have too much testosterone in your body, that testosterone makes you make less GnRH, and subsequently, downstream, you then make less testosterone. So this is a negative feedback loop. If you have too much testosterone, that testosterone inhibits this process and starts to shut down this cascade, and the testicles become a little bit smaller. If you do not have enough testosterone, then your hypothalamus produces more GnRH, which then gives you more luteinizing hormone and follicle-stimulating hormone, which then causes the testes to increase in volume. This is the primary reason why individuals who take steroids, particularly testosterone-based steroids, are known to have shrinking testicles. By artificially increasing their testosterone concentrations, they stop this process that stops follicle-stimulating hormone and luteinizing hormone from stimulating the testes. Another hormone produced by the seminiferous tubules is called inhibin. And inhibin is a hormone that inhibits this process as well. So there's a 1-2 punch. Testosterone inhibits this cascade, and inhibin inhibits the cascade. Appropriately named hormone, eh? So, guys, gals, we have a concept check now. The sperm cells are made in the blinkety-blink and matured in the blankety-blink. We have A, seminiferous tubules epididymis, B, Cowper's gland seminal vesicles, C, prostate seminal vesicles, or D, seminiferous tubules and Cowper's gland. Go ahead and rewind this video and get me an answer, or you can pause it and check your notes, check your textbook. I need an answer right now. 5, 4, 3, 2, 1. The correct answer is A. Sperm are made in the seminiferous tubules and they are matured in the epididymis. If you have any questions for me about mitosis, meiosis, or the male anatomy, please feel free to post them on the class discussion board, shoot me an email, or swing by my office when you're on campus. Happy studies!